Structure of DNA, Watson and Crick model. In my previous video, general structure of DNA was described. In this video, I will demonstrate the currently used model of DNA, which is named after two renowned scientists, Sir James Watson and Francis Crick, and the name of this model is Watson and Crick model of DNA. Helical structure of DNA, as I mentioned, was proposed by two renowned scientists, Sir James Watson and Sir Francis Crick. So after their name, this model of DNA was named as Watson and Crick model of DNA, and this is the mostly used model for DNA till date. So to say about the features of this model, I will use a color. DNA is basically of two strands. In this picture you see one strand with black color, another strand is given in red color, and these two strands are parallel to each other. The two strands are coiled around a axis. So these strands, I have taken this here, and this strand I have taken here. So these two strands are coiled around an axis. Yellow color is the axis. So these two strands are coiled helically around an axis, and the base molecules of the two strands are linked together by weak hydrogen bonds to constitute base pairs. Base molecules are adenine, guanine, cytosine or thymine. Let's say these are the base molecules here in these strands. These are the base molecules here in these strands, and over here these are the base molecules present in this strand. So this also present in this helical model also, twisted structure. Okay, so the base molecules of the two strands are linked together by weak hydrogen bonds. So there is a weak hydrogen bonds between these base molecules. Weak hydrogen bonds is present between these base molecules and constitute base pairs. Two right-handed helix are coiled about the same axis, as I mentioned, one with black color, another with red color. These two right-handed helix are coiled about the same axis given as yellow color, and the width of the DNA helix is 20 angstrom. So width is like here, it is 2.0 nanometer or 20 angstrom, and each turn of double helix contains 10 base pairs. So this is one turn, right? This is one turn up to this is one turn. So this turn of double helix contain 10 paired nucleotides. That is, this is one paired nucleotide, this is another paired nucleotide, this is another. So there are total 10 paired nucleotide in every turn of the double helix. So the double helix makes one full turn every 34 angstrom or 3.4 nanometer. So every turn, so this is one turn, and here this is one turn. So the double helix makes one full turn every 34 Armstrong or 3.4 nanometer, and the distance between two paired nucleotides, this is one paired nucleotide and this is another paired nucleotide, and the distance between two paired nucleotides is 3.4 Armstrong. And nitrogenous bases, as I mentioned in the general structure, there are 4 nitrogenous bases, actually 2 nitrogenous bases, purines and pyrimidines. Purines we have adenine, guanine, and pyrimidine, cytosine and thiamine. So adenine always pairs with thiamine by 2 hydrogen bonds, 2 weak hydrogen bonds, and guanine pairs with cytosine with 3 weak hydrogen bonds. And the 2 chains of the DNA molecule run in opposite and or anti-parallel direction. If this chain is in this direction, this chain will be in this direction. So the 2 chains of the DNA molecule run in opposite or anti-parallel directions. So the polynucleotide chains in DNA molecule show polarity. So the every strand or chain of DNA consists of nucleotides, right? Nucleotide means, as I mentioned in the general structure, it is made up of nitrogenous base, sugar and phosphate. So the polynucleotide chains, this is one chain with many nucleotides, so it is polynucleotide chains in DNA, shows polarity. Here also the polynucleotide chains in DNA molecule shows polarity. And in this point, which is the spatial arrangement of DNA, this is little bit critical. I will explain this spatial arrangement in my next video. So to describe in brief, I will introduce with this spatial arrangement. It is given in each chain the phosphate component carried by the carbon atom at position 5' of the sugar in one nucleotide unit is joined by phosphodiester bond to the hydroxyl component of the carbon atom at position 3' of the sugar in the next nucleotide chain. This is very much critical, as I mentioned earlier. So I will explain this in my next video, where I will describe the spatial arrangement of the sugar moiety, phosphate group and the nitrogenous base. For this video, this is not so important, but it is given because it is described in the Watson and Crick model of DNA. Such alternating sugar phosphate components forms the background of the DNA double helical model. So let's say this is one strand of DNA. So here will be one sugar and this will be phosphate. So this will be sugar, this will be phosphate. Again this will be sugar, this will be phosphate. Sugar, phosphate. It runs along the chain. Also in this chain, sugar, phosphate, sugar, phosphate, sugar, phosphate. You see in every chain alternating sugar phosphate components are there, and that forms the backbone of the DNA double helix model. The nitrogenous base molecules are attached with the deoxyribose sugar molecule by glycosidic bonds. So if this is sugar, here you will find, I am mentioning with green color, here you will find the nitrogenous base. Here also you will find nitrogenous base. Nitrogenous base. Here also nitrogenous base. So every nitrogenous base, which is either adenine, guanine, cytosine or thiamine, are attached with this sugar by glycosidic bond or glycosidic linkage. So to say, phosphate is there, sugar is there, and say one nitrogenous base adenine is there. So sugar and adenine, and these three together forms the nucleotide. Here I have mentioned nucleotide. So this sugar and one of the nitrogenous base is linked by a linkage called as the glycosidic linkage or the glycosidic bond. Okay, and adenine in one strand, let's say this is adenine. So this adenine of one strand combines with the thymine, as I mentioned earlier, of the other chain with 2 hydrogen bonds, and let's take this as guanine, and cytosine combines with guanine. If this is guanine, this on the opposite strands will be cytosine, and this cytosine combines with guanine of the other chains by 3 hydrogen bonds. This is the arrangement. Okay.